Hello.
Hello.
Hello. Hello. Hello.
Hi, sir.
Hi, sir.
How are you?
How are you? Fine, fine, sir. How are you, sir?
I'm good. I'm good.
I'm good. Okay, okay.
Nice to meet you here.
Yeah.
Could you, anyone hearing, could you let me also share my screen because I'm not able to share my screen now. Because or you might have to make me a co-host or something like that.
I can hear your voice, sir.
Yeah, but
Morning, sir.
oh, okay. I think I should be able to share my screen now. Yeah.
I can hear your voice, sir.
All right. Yeah. Thank you, sir.
Okay.
Could you see my screen as well?
Yeah. Now I can hear, sir.
Okay. You want me to go ahead and start? Are we good to go?
Good morning, sir.
Hello.
Good morning, madam.
Yeah.
A warm good morning to one and all. Welcome to Module 2 of Students and Faculty Development Program, Webichem 22 on the topic Drug Discovery Pipeline, the story how a drug is developed. Organized by Department of Pharmaceutical Chemistry and Analysis of Chemist College of Pharmaceutical Sciences and Research, managed by Chemist and Druggist Educational Society. And let me take a moment. The Chemist College of Pharmaceutical Sciences and Research, founded in 2006, and our sister concern, Druggist Educational Society, which aims to provide quality education through its colleges for students to gain knowledge and skills that can literally change the world. When the global pandemic halted our physical gatherings, we just found out alternative methods by opting virtual gatherings, which we call webinars. On the aim of providing most updated knowledge in our field, we, Chemist College of Pharmaceutical Sciences and Research, go for a students' faculty development program through which we conduct a series of webinars. We call it as Pharmachem or Webichem 22. So before we begin our session, let's take a moment to plead the blessings from the Almighty.
ദൈവമേ ഞാൻ നിൻ സവിധി കൂപ്പു കൈകളുമായി അണഞ്ഞിടുമ്പോൾ നീ നിൻ കൃപയിൽ തൂകണമേ. ദൈവമേ ഞാൻ നിൻ സവിധി കൂപ്പു കൈകളുമായി അണഞ്ഞിടുമ്പോൾ നീ നിൻ കൃപയിൽ തൂകണമേ. സർവജ്ഞാനത്തിൻ ഉറവിടമാം ദേവാനീ, നിന്റെ ജ്ഞാനത്താൽ എന്നെ നിറച്ചിടണമേ. ഞാൻ ചെയ്യും കർമ്മങ്ങൾ നിൻ കൃപയാലാകണമേ. എന്റെ വീഥികളിൽ നീ ദീപമായിടണമേ. ദൈവമേ ഞാൻ നിൻ സവിധി കൈകളുമായി അണഞ്ഞിടുമ്പോൾ നീ തൂകണമേ.
I invite our Honorable Principal, Professor Dr. C. Raghavan, to extend the warmth of welcome.
A very good morning to all. Respected Shri Mohanakuru PN, President, Chemist and Druggist Education Society. Respected Dr. Vinod Sitaramane, Associate Professor, Georgia Southern University, Georgia. Dear delegates, on behalf of Chemist College of Pharmaceutical Science and Research, I welcome all the delegates for the Pharmachem 22. It's an international webinar. I extend my warm welcome to Shri Mohanakuru PN President, who has given his consent to deliver the presidential address. I welcome you, sir.
Thank you.
I would like to welcome our speaker, Dr. Vinod Sitaramane, Associate Professor, Georgia Southern University, Georgia, who has accepted our invitation even after his busy schedule. He will be delivering a talk on drug discovery pipeline, the story of how a drug is developed. I welcome you, sir.
Thank you.
On the outset, I would like to thank the organizing committee, chief coordinators, Dr. John Thomas, Professor, Dr. Telly Thomas Chongon, Professor, coordinators, Mrs. Jita Amol KP, Assistant Professor, Miss Alina Jose Johnson, Lecturer, all are Department of Pharmaceutical, Chemistry and Analysis, for organizing this a very good program. Once again, I welcome one and all gathered here. Thank you.
Thank you, sir.
Thank you.
Now, I invite with great honor our patron, our mentor, and the one who gives his valuable time during his busy schedules and be with us with all his guidance and support, Mr. A.N. Mohanakuru, President, Chemist and Druggist Educational Society, for the presidential address.
Thank you. Good morning, Professor Dr. Raghavan, sir. Also, Sri Vinod Kumar Sidaramane.
Photos and thank you.
Good
Sorry,
morning.
sir. I request all the participants to please mute your microphones. I request all the participants to mute your microphones. You may continue, sir.
Okay, thank you. Again, good morning, Professor Dr. Raghavan, sir. So, Sri Vinod Kumar Sidaramane, Vice Principal, sir, Televom and faculty, and all the participants of the webinar, Pharmachem 22 International Webinar. And the topic is the drug discovery pipeline, and also the story of how a drug is developed. As a common man, I only know how to take a medicine or swallow it. I normally does not know how the effect and side effect. Because of that, the topic is of significance to the participants, particularly to the students want to know the process by which a drug is discovered or developed. I'm very happy that Principal Professor Dr. Vijayaraghavan sir has invited the art personality, Sri Vinod Kumar Sitharamane, Assistant Professor of Biology, Georgia Southern University, USA, as the main speaker. A very experienced personality was contributed so many ways to the profession, and his contributions are laudable across the world. And I understand that his CV is of 28 pages, so I'm not venturing out to read out the details about him. But I'm sure all the participants, and especially the students, will be benefited with his speech. And I'm on behalf of Chemist College of Pharmaceutical Science and Research, and on my personal behalf, and on behalf of the management, and on behalf of all the faculty and students, request Sri Vinod Kumar Sitharamane to do whatever possible for developing the institution, Chemist College of Pharmaceutical Science and Research. And one more point I just want to add up is that this webinar has got significance because the Pharmaceutical Council of India is also in the process of updating the curriculum, and they want to make it as an international level. And so at this juncture, the topic has importance, and the speaker is of immense experience. Once again, I thank the Professor Sri Vinod Kumar Sitharamane for accepting our invitation. With that, with the permission of all the participants, I declare the webinar as inaugurated. Thank you.
Thank you, sir. Today, we are privileged to have with us Dr. Vinod Kumar Sitharamane, Professor of Biology, Georgia Southern University. I'm very much honored to introduce our esteemed speaker, Dr. Vinod Kumar Sitharamane, to you all. He has done his PhD in neurosciences from University of Missouri, Columbia, USA, and he has mastered in animal biotechnology from Indian Veterinary Research Institute, Basel, and done BVSc and AH in Veterinary Sciences and Animal Husbandry from Rajiv Gandhi Institute of Veterinary Education and Research, Pondicherry. He holds a professional experience, began as a research fellow in the year 2000, followed by various positions as graduate teaching research assistant, resident, National Institute of Health postdoctoral research fellow, assistant professor, and associate professor at various renowned universities. Currently working as professor of Department of Biology, he chairs Institutional Animal Care and Use Committee, and he is a member of the graduate faculty at Georgia Southern University, Statesboro, USA. He was honored with a lot of awards, scholarships, and fellowships, in which a few among them I'm very much reluctant to leave out. He was awarded with College of Science and Mathematics Excellence in Research Award by Georgia Southern University in the year of 2019. Best Research Mentor Award by Tri Beta Student Honors Research Society in the year 2018. Society for Developmental Biology Faculty Travel Award in the year 2014. Missouri Postdoctoral Association Travel Award in the year 2012, Missouri Life Science Week Best Poster Award in the year 2011, many more and a lot more to follow. Also, he has received the Faculty Professional Developmental Committee Summer Scholarship by Georgia Southern University in the year 2015, National Institutes of Health Postdoctoral Fellowship in Developmental Neurobiology during 2009-2010, many more and a lot more to follow. He has received a lot of grants and funds from various sources like National Institutes of Health, Georgia Southern University, American Cancer Society, and various sources from altogether worth more than $2 million, which approximately over 20 crore Indian rupees. He has more than 30 peer-reviewed publications and more than 70 selected refer conference publications in his account. He has guided and supervised numerous UG research scholars and received a lot of student research awards, altogether worth more than $128,000. I wonder about the joys and nights might have been sacrificed to you, sir. With due respect, once again, I invite Dr. Vinod Kumar Sitramne for his session for Module 2 of Students and Faculty Development Program, Webichem 22.
Thank you, everyone. Thank you, Mr. President. Thank you, Mr. Principal, Vice Principal, coordinators, and all the faculty and students for inviting me to be here. I'm more than happy to talk to you. Hopefully, I'm able to give you something that you can take with you. And thank you again for a generous introduction. Before I proceed, I just want to make sure you guys are able to hear me okay?
Yes, sir, you're audible.
All right, perfect. Thank you. So yeah, today I'm going to be talking to you about a topic that's drug discovery pipeline, the story of how new drugs are developed. So before I get into the topic, I want to briefly tell you where I come from, and then I'll get into the topic and tell you a few things that we've been doing in my lab. So the picture that you see here is the Georgia Southern University. It's a beautiful university. It's around 120 years old. It's one of the flagship universities in the state of Georgia. So if you're wondering where it is, this is the United States map, and this is the state of Georgia right here. And off the coast of Georgia is the Georgia Southern University. This is where I am. I've been there for 10 years. Before that, I was in Missouri, and before that, I was in India. I'm basically from Trichy, Tamil Nadu, where I did all my schoolings, and I went to college in Pondicherry, and I did my master's in Delhi. So that's been my life in general, So I've been in the States for almost 20 plus years now, and I still miss India. There is nothing that compares home. I have to be honest with you. And, I've been here for a month now, and I have to leave next week, and I'm not feeling it yet. So it's just crazy how things work. No matter where you go, you always miss home. Anyways, so what I want to do today is this is what I have in mind. So I'm not sure if we're going to have time to get through all of this, but I want to briefly, this is the outline of what I'm going to be talking. So I want to talk about drug development pathway. Basically, I want to tell you how a drug comes into existence and what are the different steps involved. A lot of you may know parts of it, but I'm going to tell you the story from the perspective of how things are done in the US, and I'll just compare how things are done in India, but for the most part, it's very similar. And then I want to talk a little bit about some of the things that we do in my lab, which is we are trying to identify drugs and novel therapeutic strategies to treat cancer. Believe it or not, this is one of the most important diseases of our lifetime. So, so that has been one of the focus in my lab. And I'm going to talk a little bit about what is cancer, what are some of the potential targets. And I'm also going to talk about some of the strategies that we have developed in our lab that includes gene therapy and chemotherapy, identifying new drugs, also using cancer nanomedicine, like basically using nanomaterial-based therapies for cancer. And then I'm going to summarize and conclude over there. So I do want to point out that, some of these could be a little off or deep for you guys. I'm going to try to skip the details and tell you the big picture stories as much as possible. And I would like to keep it as interactive as possible. So if you guys want to talk or raise questions in the middle, please feel free to do that. I'm also going to try to keep some time towards the end where you can ask me questions as well. All right. So let's go. So what is the drugs? I'm not sure what this, are you guys seeing this line as well, the yellow line?
Yes, sir.
I don't know where it came from, but that's okay. So what is a drug? So it's important to know what a drug is before we go on and talk about how drugs are made, how it's coming into existence, how it comes to the market. So I'm going to give you a broad definition of a drug. A drug is defined as a substance which is either chemically, biologically, or biotechnologically originated. A drug, we need to know the structure of the substance, whatever it is, we need to know what the structure is. And it needs to be something other than a nutrient or an essential dietary ingredient. So bottom line, this is a chemically, biologically, or biotechnologically originated material, which when administered to a living organism, it produces a biological effect. And most of the time we are using this to either diagnose, prevent, or treat a particular disease condition. And that's how we define a drug. So in the past, it used to be just a chemical, but it's been expanded to include biological as well as biotechnologically originated material. And again, like I said, in the past, drugs were known, we used to call drug discovery, but now it's more of a drug invention, because nowadays drugs are being designed and made. In the past, it used to be discovered from natural products like plants and other materials, but now it's no longer discovered. I mean, it's still discovered, but majority of the drugs which are coming to market are designed by humans. So it's more of a drug invention. So how does a drug come into existence? So if you think about it, a molecule or a material or substance need to be invented with a particular purpose of treating, having a particular effect, or treating or diagnosing a particular condition. So that's one major step, which is the discovery step, identifying and designing a molecule or a substance or a material. Number two, we need to develop it. You can't just take any material and just give it to humans or any other animals and use it as a drug, because all these materials, if you might have heard this saying, everything is a poison, every food is a poison. It only depends on how much you take. So likewise, every drug can have its own effect and toxicity. So a drug or a material needs to be developed. So that's why there's a lot of developmental principles involved. Broadly, we put that into two categories, which is preclinical and clinical. So I will briefly go over what that is in a minute. So bottom line, a drug needs to be invented and it needs to be developed before we can take it to what is called approval. So these are the major steps. If you look at it, the step one is basic research, early discovery, and preclinical. And a lot of it is done in vitro or in vivo. Again, in vitro or in vivo meaning it's either done in the lab in plastic materials, or cell culture done in the lab without using animals or using bodies, or in vivo. Nowadays, a lot of these drug discovery are also done in vivo. And a lot of our studies are done in vivo too. I'm going to show you what that means, using animal bodies or living animals. So first step is basic drug discovery. Then it goes to human testing. Once you find these drugs to be suitable with less toxicity and with potential efficacy, then it goes to human testing. Before it goes to human testing, there is an extensive application process involved. It's called investigative new drug application. That's what IND application means. You have to create an application and you apply to the regulatory agencies. In the US, it's called FDA. In India, we have what is called Central Drug Standards Control Organization. So likewise, every country has its own organization that controls this drug discovery and processes. So before any material or drug can go into human testing, it needs to go through an extensive application process. Then it moves into human testing. And within human testing, there are three phases: phase 1, phase 2, and phase 3. I'm going to tell you what those are in a minute. Once we have acceptable results, or results that is valid and good to go, then the company or the person who's developing it would apply for what is called a new drug application. And again, it goes through the regulatory authorities. Then the authorities will review these applications, will look at all the data, and this review process typically takes a year or so. And then it will get approved. even after it gets approved, it's not the end of it. All these regulatory agencies will actually market and surveillance, do a survey of post-market monitoring. Typically, let's say it's given to humans, they are going to collect data from humans, patients, as they take these drugs, and they constantly make improvements. And if they have to make changes to the approval, they will so. So that's the overview of the entire drug development pathway. So bottom line, you have an early discovery step, and then human testing, and then it goes to data review and approval. And then after approval, once it's in the market, it still get completely surveyed. So it's still the approval agencies are collecting data to assess the efficacy and toxicity as it happens, because sometimes the testing may not be good enough. All right, so let's quickly see what all happens in each one of these steps. So step one, like I said, which is the discovery step. And what happens here is typically the investigator or the company that's developing the drug, they would have a particular disease or a target in mind. So for example, if someone wants to develop a drug towards diabetes, so diabetes is the target. So they're going to look at different pathways involved in diabetes. What are some of the molecules you can target so you can control diabetes or the sugar level or different aspects of diabetes? So that's usually going to be the targets. So, and then they're going to use a set of molecules. Let's say if you want to find a new drug, and typically these companies, the big companies, they usually start with 15,000 to 20,000 molecules. So there's a huge chemical libraries they start with, and they go through these molecules with assays. They would have specific assays to test if these drugs are affecting the particular pathways. And if they do, that's what we call it validation. They would say, okay, here is a drug in the 20,000, there's 200 drugs that are validated. The remaining, let's say 19,800 drugs are not validated. So they're going to eliminate all those different drugs that's not going to be useful. And with the 200 that they have, they're going to refine it. They're going to say, okay, let me see these 200. Do they all look good? Do they all have the same efficacy? They're going to find the ones which is the best efficient, which means which works the best. And they're going to find the drug that has the least amount of toxicity, because all these drugs might have other toxicities. So I'm going to talk about that too. So they're going to refine it. That's what we call lead optimization. So it's all, in other words, it's called lead refinement too. Bottom line, they're going to optimize and find, narrow down the 200 to let's say 10 drugs. Once they have the 10 best leads, then they're going to take it to preclinical development. That's when they use animals. This is where they use lab animals, and typically before a drug can come to human studies, a drug needs to be tested in non-mammalian models. That means you might have to use like fish or birds, something like that. And then you'd have to test it in mammalian models like mouse or rat. And then you would have to test them in higher vertebrate, higher mammalian models like dogs or primates before it can get to a human. So there is a huge amount of work in the preclinical development phase. So you can't get to a human study before all of these phases are completed. So it takes a lot of time. So I'm going to give you a idea of how long it takes to. So let me tell you what else happens in step one. So again, we're still talking about step one. So the target identification, validation, then they're going to go to what is called hit to lead or lead optimization, which is when they're going to find the best possible drug with the least amount of toxicity and best efficiency. And then they take it to preclinical studies. And here's where two major things happen. And some of you might have heard of these two things, which is pharmacokinetics and pharmacodynamics. This is when they are studying how or what the body does to a drug, which is what we call a pharmacokinetics, and also what a drug does to the body. That's what we call as pharmacodynamics. So bottom line, these studies will help us understand a lot of things. For example, nowadays a lot of drugs are produced, so the drugs can be slowly released, which is what we call as long-term drugs. You can take a drug and that drug will slowly release over a period of time. So it kind of reduces your need to take multiple doses. So some of these pharmacokinetics involves, okay, how long does the drug take to get liberated in the body? And once it gets liberated, when does it get absorbed? How much does it get absorbed? Where does it go? That means distribution within your body. And how does it get metabolized? That means how does the drug get broken down and what happens to these broken down pieces? And does it get eliminated? And how does it get eliminated? How long does it take to get eliminated? And where does it get eliminated? All of that is what the body does to the drug. So we need to know every single detail before it can go to humans. All of this is done in animal studies. And the next step is pharmacodynamics, which is what a drug does to the body, right? So like I said, any drug, anything that we eat could be poison. It all depends on the amount of we are taking. And every drug has its own specific mechanism. Some drugs could be cardiovascular toxic, meaning it can affect your circulatory system, your heart and vascular system. Some drugs could be affecting your brain. Some drugs could be affecting your hormones. Some drugs could be affecting your kidneys. So we need to know every single detail about every single piece of the drug. After it gets broken down, what all happens to each one of these pieces. So a lot of details need to be figured out, and all of that is done using preclinical studies. That's what happens in preclinical studies. And why is it important? Because all this information needs to go in the application for taking the drug to human studies. That's what I said as investigational new drug application. If you want to apply for human studies, you have to put in all these details, and the regulatory authorities are going to look for all these details. They're not going to let any drug go to human studies without any of these details. So that's why you need to get all these details before it can go to humans. So once it goes to humans, let's say you got collected all these details, you got, a good amount of drug and, good, there's not much toxicity, but we still don't know if it's toxic for humans. So that's why it goes into a clinical development, which is where all these drugs would get studied in humans. And this is where there are three phases. There's phase 1, phase 2, and phase 3. So phase 1 is a simpler study. It uses very low number of humans. Typically, there's only 20 to 100 healthy individuals or individuals with the condition. So again, this is the phase 1 of human study, because this is where they're going to look to see if this drug is safe for human use. They don't want to use it on 1,000 people and find out that it's not safe. So they're going to take very little amount of people, let's say 50 to 100, and then they're going to look to see if this drug is safe and what kind of dose to use, because again, dosage matters. Again, when I say dosage, I'm talking about, do we want to give, let's say 100 gram or 200 gram? Do we want to give it 2 times a day or 3 times a day or 1 time a day? All those will be figured out in phase 1. Once it is safe and it's not toxic, then it moves on to phase 2. And again, before we can move on to each of these phases, the regulatory authority needs to approve so you can go on to the next phase. So they're going to look at the data and follow the data at every step of the way. So then in phase 2, here's where the numbers increase. At this point, they're going to actually look to see if the drug does what it's supposed to do. Meaning, if the drug is supposed to cure a disease, now is where they're going to see if the drug actually does it or not. So now, typically, this is where they would use around 1,000 people. And if it's a rare disease where there's very little number of diseases occurs, then they're okay with 50 to 100 people. But in general, for a common disease, they would want to have at least around 1,000 people. And like I said, this is where they're going to look at the efficacy, whether the drug is doing what it's supposed to do. And they're also going to look at the side effects. And again, this is around 1,000 people. Now, if it's positive, and if it's good, if it's a good result, meaning it is efficient, and if it has less side effects, then it moves on to phase 3, which involves a large number of patients. This is where it typically goes to, again, tens of thousands of people. For example, for COVID vaccines, they had around 30,000 individuals around phase 3. So around phase 3, you need tens of thousands of people to be tested at that time. And again, they're going to collect all the efficacy and side effects at that point too. And with that data, that's when they're going to really assess the benefit versus risk, meaning, is the drug really beneficial to cure whatever the condition is? Is it really going to do? Or is there any risk? What is the risk versus benefit ratio? If it's more risk, then it's not worth to go through all this process. And like I said, the regulatory authority is going to be looking at the data and following this data at every step of the way. And they have the ability to stop and, any of these phases at any point of time during these trials. And who does all these clinical trials? Typically, it's done by the companies which is developing the drug, or it can be done by hospitals if they are developing drugs. So it could be either of those. Or if an individual is developing a drug, then they could be part of the company and they could do that too. So it could be any of these entities that could be doing all of this, but before they can do, they have to get the approval from the regulatory authorities like FDA in the US or CDSCO in India. So once it goes through all these clinical trials, that's step 2. Then we move on to the final step, which is the data review. So this is where all these data that's been collected goes back to the regulatory authorities with a new application that's called new drug application. So again, all of this information will be put in this application and sent in, and the regulatory authorities typically take around a year, 10 months to a year, to go through all these data. And it's not done only by the scientists. It involves almost everyone from the society, like, regular common person or a scientist, mathematician, biologist, physician, everyone is involved, and a lot of information is passed through, and everyone gets to look at the data. And finally, they get to vote based on the success of the results. And that's when it gets approval. And if it gets approval, then the drug gets registered. But, like I said, that's not the end of the story. That's when it moves on to post-market monitoring, meaning there is a reporting system. Usually there is a data analysis system where anyone can report. If someone is taking the drug, they can report any potential adverse effects that they might be going through. And all of this is part of post-market monitoring. And again, the regulatory authority will continue to monitor these drugs throughout the life of the drug. So that's what they are supposed to do. They will monitor the safety even after the drug is approved. So again, these regulatory authorities have the ability to go back and say, hey, something is not right. People are complaining. We need to go back to this testing again, or we need to, cancel or rescind your approval. Whatever it is that needs to be done, they would do it. So in general, hopefully that I kind of gave you an overview of how these things work, how a drug is developed and how it gets to the market. And even after it's in the market, it's not the end of the story. It continuously gets monitored. So there is early discovery phase, and there is a clinical development, and there is a data review and surveillance phase. And an important thing here is it's a huge process. It is not a trivial process. I want to give you an idea of how long it takes and how it gets into existence. So like I said, typically for every one drug that gets approved, if you look at the starting drug discovery phase, 15,000 compounds get screened. And the preclinical around 500, a lot of it gets thrown out, 500 comes in there. And out of that, only 10 gets to clinical trial, which is where human studies are done. Out of that, only 1 would typically get approved. And you could imagine this is a long process. And for any one drug that gets approved, typically it takes around 15 plus years. This is not a trivial time. You guys must understand time is money. For every drug, a single drug, it takes around 15 years. And it also costs around $2.6 billion on average. You know why, and these drug companies are wanting to make so much money, and they're putting a lot of effort. We can't blame them, but still, it's a lot of money to go through all of that to find a good treatment. So that's why it's a high risk, high cost business. They have to go through all of that, and finally, after a long period of time, you might find a good drug. And there's 12% chance. For every 5 compounds that goes into FDA for approval, there is 12% chance that it might get approved. And I also want to give you an idea of how many drugs get approved every year. You can see every year around 500 or 600 applications are received in FDA, but only around 50 or so gets approved. And a lot of these approvals are not, broad spectrum approvals. They would have very specific restrictions. Okay, you can only give it to this type of individuals. You can only do this. So there's a lot of restrictions that come with these approvals. But either way, you could see the cost and the time involved in getting one drug to the market. It's very, very stringent, very rigorous process to get a drug out in the market. And again, after all of that, we still find some drugs that's not working right, and there's a lot of side effects and things like that. That's just the way it goes. But one thing that a lot of people are trying to do here is to reduce the cost involved and also the time involved in this process. If you go back to what I've been telling you, the cost and time involved primarily are at this phase. You can see the discovery phase and the preclinical phase is typically where you take around 15 or 13 to 14 years goes in this phase. So people have been trying to cut down the time here because time is money. Again, people have been trying to cut down the money here. What people have been doing is developing computational methods, computer-aided drug discovery. Bottom line, they're developing algorithms, or what we call as AIs, artificial intelligence or machine learning driven drug discovery process. Bottom line, you put an input, a drug, a structure, the computer, the software would be able to analyze the structure and say, hey, looks like this molecules will bind to this kind of proteins in your body. So these are potential targets. These are not good targets, so you can remove them from the pool. So it basically helps you to cut down on the time and the money spent on all these drugs. Likewise, another major aspect is preclinical aspect, pharmacodynamics and pharmacokinetics, what the body does to the drug and what the drug does to the body. Again, people have been developing algorithms, computer-based softwares to predict what a drug would do to your body, what a drug would do to your specific organ systems, is your, let's say, your nervous system or a urinary system or a kidney, or what the drug would do based on the structure of the drug. And for the most part, a lot of these algorithms have been pretty good. And again, these are getting refined, the better algorithms are being developed. The whole idea of this is to cut down the time and money involved in the early discovery phases. And it's been very useful, but again, there's not been any compromise on the safety. Even after all of that, if we come down to, let's say, 100 molecules, again, every one of them would go through the preclinical and the clinical development to be sure that it actually is safe to use. So the idea is to save time at the early part of discovery. That's where the idea is. All right. So with that, I'm gonna give you a moment to quickly ask me any questions before I go on and tell you about what we do in my lab. If you have any questions, please feel free to ask any questions. because I've been talking a lot. Any questions? All right, so I'll just continue. So maybe I'll just give you another few minutes and I'll talk about what we do in my lab for a few minutes, and then I'll open the floor for questions. All right, so let me just quickly move on to what we do in my lab. So my lab, again, we've been doing a lot of early drug discovery. That's where new molecules are getting discovered. So we collaborate with a lot of chemists and other physicians to discover new drugs, especially for cancer. So I'm going to quickly talk about cancer, and I'll tell you one of the stories of what we've been doing, and then I'll just complete my talk at that point. So what is cancer? I want to quickly give you an idea of what cancer is. If you look at cancer, so here's an animation of what a cancer is. Our regular body cells, when they accumulate mutations, so you can see these cells which are turning a little green, again, that's just pointing out to accumulation of mutations. When I say mutations, what I mean by that is changes in the DNA, the genetic sequences that make these cells proliferate, meaning they start dividing more and they start growing. And if they're just growing, it's what we call as benign tumor. They're staying within the limits. It's all good for the most part. It doesn't kill us. But when a cancer becomes malignant, that's when it starts being more dangerous. And when does the cancer becomes malignant is when these cancer mass, you can see this mass of cells growing, they start attracting blood vessels. You see all these brown things, these are the blood vessels, red things are blood vessels, or body blood vessels. Once these blood vessels are, these cells are growing, they're going to need food, nutrition. So they're going to start getting this blood from our body so they can start growing. Basically, they hijack our body's nutrients to help them grow at the expense of other normal cells. So that's the problem here. So here's all the blood vessels from the body that's supplying nutrients to the cancer tissue. Once they do that, again, it's still okay. But what happens is some of these cancer cells, you'll see this green cell right here that's getting into the blood vessel. These are what we call as metastases. These cancer cells will start getting into these blood vessels and they escape to a different region. And they go to what is called secondary sites and they start growing at the secondary sites. That's when it becomes a malignant cancer. And majority of the death or mortality in humans as a result of cancer is because of malignancy. Malignancy is the major reason for mortality. And believe it or not, cancer kills around 10 million people every year, which is a lot of people. And a lot of these could be saved if we do the proper prevention and treatment. Another really important aspect of cancer is the economic aspect, which is around $1.16 trillion. Again, just to give you an idea, the GDP, Indian GDP is 3.5 trillion. So cancer economic impact is almost half the country's GDP. That's a whole bunch of money involved. And cancer is being considered as a group of diseases. I mean, in the past, we used to think it's one disease, but now we know it's a lot of diseases together because lots of different mutations contribute to it. And in humans, major cancers are the prostate and breast cancers are the leading cancers in men and women. And but the major cancer that leads to death is the lung cancer. So obviously cancer is important, a lot of economic as well as lives are involved. So it's important to cure them. What's being done and what can be done better? Traditional therapies. Nowadays, you might have heard of a lot of traditional therapies, chemotherapies, as well as ablation-based, radiation-based, laser-based therapies or surgery-based therapies. A lot of these are good, especially if they kind of get them early on. but as it gets, to the later stages, these cancers are really hard to control. The problem is, it's hard to diagnose cancer. We don't really see the symptoms until you actually get to the malignancy stage where the cancer is already spreading. So either we have to improve the diagnostic process, or we got to start getting better treatment procedures to prevent these cancers. So one of the major goals of cancer therapy is to increase the specificity and to decrease toxicity, because a lot of these radiations, you might have heard, they have a lot of side effects. A lot of chemotherapy, they have a lot of side effects. You might have heard like people losing hair, a lot of skin issues, a lot of digestive issues, because a lot of this chemotherapy kills proliferating cells. And we have a lot of these proliferating cells in the digestive tract, skin, hair, and a lot of these places. So they are not specific. That's why you start to get a lot of toxicities with that too. So what we've been doing is we've been developing some of the drugs which could be a little bit more specific. We are trying to find some gene therapies, bottom line, knock down or delete specific genes, and then which can lead to complete prevention of metastasis, or stopping the spread of cancer. We've also been developing some drugs to stop the spread of cancer. We have also been doing some nanomaterials, but hopefully, maybe I won't be able to talk about the nanomaterial today, but I'll just quickly talk about this gene therapy and chemotherapy briefly. So what have we been doing? Just to give you an idea of what happens in cancer, I showed you the animation, but I'm just going to quickly show you another way of animating here. So let's just imagine these are your normal cells in your body. What happens is these cells, some of these cells will start getting mutations. So the red dot that you see is one mutation, but they start accumulating mutations. That cell starts getting 2, 3, 4 mutations. Once they reach a what is called threshold number of mutations, the cells will start proliferating like crazy. More cells start coming out. Once cells start coming out, now they're going to start knocking down the boundaries. Typically, any tissue we have in our body, the tissues have boundaries. We have what is called extracellular matrix. These are nothing but proteins around these tissues. These are boundaries in our body. But if these cells need to spread out, they have to break out of these boundaries. How do they do that? The best way they can get out is through the blood vessels. So what these cancer cells do is they start mimicking or hijacking our body. What they do is they start producing molecules. You see these blue dots in here? These are some of the molecules that the cancer cells would secrete. These molecules will interact with the matrix, and they will interact with the blood vessels nearby, and they will kind of break open the matrix, and they will allow for the blood vessels to grow into the cancer tissue. And then some of these cells will hop into the blood vessels, and they will move out of these tissues. This is the primary side. This is the primary side. They will move out to a secondary side. A lot of these cells will come out, and some of them will get into the secondary side and will start growing into a new tumor. And that's what happens in cancer growth. One of the best things to do is to prevent cancer is to block this process, especially the spread of cancer is this. If we can block the metastasis process, somehow block these cancer cells from going from the primary side to the secondary side, that is the best thing to do, because 90% of the mortality is because of tumor spreading. Majority of the human dies because of malignancy, because of tumor spreading to other sites. So if we can somehow block it, that'll be the best thing to do. And all of this is happening because the environment of the tumor is being modified, because these cancer cells will start to secrete some molecules that changes the environment. So we need to know what's happening with the environment. So one of the things that we found out is a molecule called, again, I'm going to skip over the details here. There is a protein that's on the cancer cell called integrins. So these are the proteins that's on the cancer cells. What they do is, in a cancer, in majority of the human cancers, this protein would get upregulated, meaning a lot of this protein is produced. And that helps this cancer to grow from one side to other side to spread and cause death. So if we can somehow block this protein, we might be able to block the spreading of tumor. So what we wanted to do is find a way to block this protein, and we wanted to test it. So what we did is we first tested what does this protein do. I'll skip the details and tell you what the protein does. This protein, integrin alpha 6, what it does is it allows for new blood vessels to grow into cancer, because that's how cancer can get into the blood vessels and go into other tissues. And it allows for cells to spread to other parts of the body. So basically, this protein allows for metastasis. So we wanted to block this protein being produced in the cancer. And we wanted to test if that helps. So what we did is we used a fish model. So again, a lot of our studies are done using zebrafish. This is an amazing model. Some of you may have heard about zebrafish. Actually, this is a fish that originally was discovered in India, but it's getting wide usage all over the world for drug discovery and biomedical applications. And we use this fish to study too. The good thing about this zebrafish, again, they have lines in them. That's why it's called zebrafish. It's got conserved targets. Humans and fish have similar proteins, similar mechanisms. They also metabolize the drug very similarly. Physiology between humans and fish is very similar. So a lot of things are conserved. So if we can develop a drug in fish, you could easily translate it in humans. So that's why we are using zebrafish to test some of these therapeutic strategies. And what we do is we use these embryos, fish embryos. We can easily breed these fish, like basically put males and females together. They usually lay hundreds of eggs, and we can put them in plates, and we could have like 100 well plates or 384 well plates. We can put one embryos in each well, and you could have different molecule in each well, meaning you could screen like 300 drugs in one day. That's like super high throughput. That's like quick, very quick compared to any other methods. You can't even imagine doing these in rats and mice. And it's costs so much more money to produce rats and mice. And let's say if you're breeding a rat, a male and female would typically produce 8 to 9 babies. But these guys would produce 200 to 300 babies, embryos, so you can actually screen them much more faster with these fish model. So really quickly, what we do is here are the fish. You can see these. We put these males and females together in a tank like this, and they will start laying eggs. You will start seeing eggs coming out of here. You see this one little dot that's going down, that's one embryo. And soon you're going to see a lot of these embryos starting to come down, and they'll get collected at the bottom. You could see some of these eggs coming down right there. All these are fish embryos that gets laid, and you'll get all these embryos collected at the bottom of the tank. So it's that simple. You can collect like 100 to couple of 100 eggs in any given day. You get these eggs, and then what you do is you basically let them grow. And it grows really fast too. This is what an egg looks like as soon as they're born. Within 5 days, they will look like a complete fish with every organ you could imagine. There's got heart, liver, pancreas, kidneys, everything is there by 5 days. And all of them are functional. So they're essentially, you can test all of that functionality within 5 days. And the good thing is they do not have an immune system by then. So what we can do is we can actually put human cancer cells into these fish. So we use some of these transgenic fish where we can visualize the fish blood vessels. What you see in green are the fish blood vessels. We can literally see every single blood vessel. And what we do is we put human cancer cells into the fish. Here's how it works. This is what we call it. We call it as humanized zebrafish tumor xenografts, meaning we put human cells into fish. So we'll take human fish embryos. We inject them with what is called antisense nucleotides, which will block the expression of the integrin that I talked about. We wanted to block the gene. Then we take human prostate cancer cells, we label them, we put a dye into these cells. And these are live cells, by the way, and these are live fish, by the way. And we inject these fish with the human cells. So it's human, it's a fish, where we put human cancer into them. And the human cancer grows in the fish because fish at this stage do not have an immune system. So it allows for a human cancer to grow. And by 5 days, the cancer grows and will start to spread. So now we would ask whether if having this gene knocked out will block the spread and growth of the tumor, human tumor in the fish or not. And if it blocks, that's what we want. If it blocks, then we have a therapy at hand. Then we could translate to humans from there. And I'll just quickly tell you what we did. So here is a fish. You could see a fish body. All these green things that you see are the blood vessels in the fish. Now, you do not see any cancer cells in there. Now, if I put cancer cells, you'll see red cells in here. If I put cancer cells, here is a fish with the cancer cells. Again, these are human cancer, prostate cancer cells into the fish. Now, one thing I want you to see here is you see this green blood vessels growing into the tumor. So bottom line, if you see if there is no cancer cells, the blood vessels are not going to grow here. But if there is cancer cells, the blood vessels are going to grow here. Basically, the cancer cells are attracting blood vessels to grow into the tumor. Why do they do that? Because now they can spread. You can see if we put these cells here, now the cells are starting to spread. You could see these red cells going towards the tail of the fish. And here is the blood vessels. You can see these red cells getting into the blood vessels, and they're going in the blood vessels and moving into the blood vessels. Now, that's the cool thing. This is when integrin is present. Now, when I take away the integrin, when I delete the integrin with this gene therapy, where we block the expression of the human integrin, what happens is this tumor does not spread. It remains in the same region. It cannot go anywhere because no blood vessels growing into this tumor, and you do not see any tumor cells into the blood vessels. Now, that's very simple. Now, I've skipped over a lot of the details here just to give you the important things. So what we found out here is essentially when we delete the gene, we literally reduce and completely reduce the ability of new blood vessels to grow and the ability of the cells to metastasize. Essentially, what we think is happening is blocking this gene completely reduces the metastatic process and metastasis of tumor, and that can save a lot of people. And this can be something of a preventative. People can take this as a preventative measure, like, you know, when you turn 50 or so, people can start taking this, and that can block, even if someone has a tumor, that can block any tumor from metastasizing, and that can help people a lot. So that's the gene therapy that we've been developing. One more thing I want to quickly tell you is, you know, we've been trying to develop other drug-based therapy to block metastasis as well. And for that, what we do is we screen drugs. This is where we use the computational method, where we obtained what is called a chemical library. You can get a library of chemical drugs. We obtained like around 1,500 drugs, which is a library of chemicals. And we start treating these fish with tumor with these drugs. And we looked for fish where the drug would block the metastasis. Basically, it will stop the spread of the tumor. And here's where we use some of the computational method. So what we did is a computer-based assay where we used the structure of the drug. We input the structure of the drug in a software, and the software predicts only 60 of these compounds will be active towards a particular protein, because we wanted to block a particular protein called VASF3. That protein is also involved in metastasis in humans. So we put the structure of the protein, we put the structure of the chemicals, and we asked the computer to predict how many of these 1,500 would be useful. The computer predicted only 60 of those would be useful. And we took the 60 of those and we did the screening with the zebrafish xenografts. And what we did find is only 3 of those to be valuable in blocking the metastasis. Here's what happened. So literally, I'm going to show you the 3 drugs that we discovered. Here are the 3 drugs. I'm showing you the structure of these drugs. I'm showing you the binding, the algorithm that allows for testing the structure binding to the protein. So this is the drug that's binding to the protein here. These drugs would bind to the protein. So, and then we tested using the xenografts. Here's what we found. So normally cancer cells are very invasive, meaning they would have a lot of protrusions. They would like to move a lot. So they're going to have a lot of protrusions. They will move a lot. But when we treat these cancer cells with the drug, you can see all these lines here. That's what we call as protrusion. These are cancer cells, normal one. So, but when we treat with the drug, you can see how the line, all these protrusions are gone. So the cells are not able to move anymore. In fact, we can see these cells not able to migrate at all. You can see this when we treat with the drug, the cells have become more round instead of being spindle. So the drug actually works. The drug is actually making these cells less mobile, invasive, or metastatic. So we also tested in the fish. So here's what here's a live animal. You can see we put these cells, cancer cells, on a fish. You can see these cells moving. So this is a video of cells moving inside a fish. All these green cells you see, those are cancer cells, human cancer cells that's moving inside a fish. So now what we do is when we treated these xenografts with the drug. So here is a normal one. You can see the tumor cells and they are spreading all over the fish. You can see these red dots all over the body because it's metastasizing. Now, when I treat this fish with the drug, you can see this cancer is restricted to the same place. It cannot even spread. Again, another wonderful drug that can block the metastasis and that can help save a lot of people if it's used as a preventative measure. So again, I think hopefully what I've done here is just quickly, what I've tried to tell you here is I've given you an overview of drug development pathway, how early drug discovery leads to what is called preclinical and clinical development. And then based on the data, it goes on to become a review process and gets approved and also goes to constant monitoring or surveillance of these drugs that's on the market. That's the big picture of how drugs come into market. And I've also given you an overview of what we do in my lab, developing new drugs, both gene therapy and chemotherapy based methods to develop new drugs against cancer. I do not have time to talk about some of the nanomaterials that we've been developing, so I'll just skip over those and I'll just quickly acknowledge all the work that's done by the students, because all of these are done by students. I pretty much just help the students, give some ideas and things, but a lot of it is done primarily done by the students and the funding we've been receiving to do these. And a lot of these are undergraduate and graduate students. And I do want to point out that if any of you students ever want to work with us, please reach out to me. I'm more than happy to help you either find an opportunity with us or other, I have a lot of friends all over the States and all over the world who are happy to work with you guys. So please do reach out. I'll be more than happy to help out. And all you have to do is just look up my name, Vinod Sitaraman. If you Google, you'll find my lab and all those details, or I'll leave my details with the your faculty and coordinators. They might be able to help you too. So with that, I'll stop right here. And please, I'm sorry if I went over. And thank you for the opportunity. And thank you again, Mr. President, Mr. Vice Principal, Principal, coordinators. Hopefully, I've given you something to think about. I'll be happy to take any questions here.
Thank you, sir. Now the session is open for interaction and we will take some time for queries now. Participants may drop your queries into the chat box of our Zoom meeting or into the comment section of our YouTube stream, or you can shoot your questions by unmuting your microphones. So meanwhile, shall I ask you a question, sir? Actually,
Yes,
it is related to the first part of your session, that
Yeah.
whether the clinical trials are done for Asian humans. If yes, whether it is done for all the drugs. If not, how a drug that has been tested in a different community can be effective for we Asians having a different genetic distribution.
Yes, absolutely. That's a very important question. That's a great question. Yes, any drug, when it comes to clinical trials, one of the considerations that's being done is, one is what type of condition that's supposed to be curing. So let's say if it's a particular drug condition like diabetes, so they're going to look to see the racial or ethnic distribution of the particular disease. And depending on the disease, let's say if this is a particular condition that's more widespread in Asian or African Americans or Caucasians, then the regulatory authority is going to want to have a population with appropriate ratios before they can approve for a broad spectrum approval. So that is something that's taken care of at the regulatory level. And that is something that they do look for. Let's say the investigators or the clinical trials managers, they are looking to recruit people of different ethnicity to be able to come to a broader conclusion. So yes, that's something that's definitely considered while doing the clinical trials.
Thank you, sir. So we are having a question from the chat box from Aishwarya Lakshmi, that any in vitro method to study inhibition of integrin protein?
Yes, there are plenty of methods. A lot of these can also be done in cell culture. So, so some of the things I have not shared is the data. All of these studies that we have done, we usually first do it in the in vitro, like in the cell cultures. We block these proteins in cell culture. But the more important thing is in vitro is very different from in vivo. In vitro should lead to in vivo, because in vivo is a dynamic environment, the physiological environment, metabolism happens in vivo. So drugs, lifestyle or life of drug is completely different in vivo. So in vitro leads to in vivo. I don't know if I answered your question, Aishwarya. That's a good question.
Thank you, sir. So it's looked like that we have covered all the queries from the audience.
Okay.
So is there anything else you want to cover before we winding up the session?
No, thank you again for the opportunity. Hopefully you guys got something out of it. Hopefully I didn't take too much time. Please do pass on my contact details to students if they are interested in talking to me. I'm more than happy to work with them.
Sure.
And I'm happy to work with you guys as well. Please be in touch.
Thank you, sir, for the cordial invitation for our students.
Thank you.
So I express my deep appreciation and sincere gratitude to Dr. Vinod Kumar Sitramani for the wonderful and informative session. The link for feedback has been shared to the chat box of our Zoom meeting and the comment section of your YouTube stream. And I kindly request all the participants to fill the feedback form to receive your e-certificates. So I'm sure that our participants have gained a blend of knowledge in the field of drug discovery, drug development, clinical trials, and about the cancer growth and development. Now I invite Mrs. Jidamol KP, Assistant Professor, Department of Pharmaceutical Chemistry and Analysis, to deliver the vote of thanks.
A very good morning to all. I deem it a great honor and privilege to have the opportunity to present a vote of thanks for this webinar. First and foremost, I thank our resource person, Dr. Vinod Kumar Sitaramani, Professor of Biology, Georgian Southern University, Georgia, for enlightening us with his. I extend our heartfelt thanks to Mr. A.N. Mohanakur, President, CDS, for his support. We are extremely thankful our principal, Professor Dr. C. Vijayaraghavan, for his enormous support and guidance. Thank you, sir, for being with us. I would like to thank our chief coordinators, Dr. John Thomas, Dr. Tony Thomas Chungat, for their valuable guidance and support. I owe special gratitude to all our colleagues who always stand with us and motivate us. I thank participants, System Administrator Mr. L. Jacob, my teammate Alina Rose Johnson, and last but not the least, our students for making this webinar a grand success. Once again, thank you.
Thank you.
Thank you, ma'am. So on behalf of CCPSR family and everyone present, I would like to thank Dr. Vinod Kumar Sitaramani for his significant contribution to Module 2 of Students and Faculty Development Program, Webichem 22. Also, I extend my sincere gratitude to all the delegates present, and until we meet again for Module 3 of Students and Faculty Development Program, Webichem 22, this is Alina Rose Johnson signing off. Thank you and good day to you all.